Right, we're back for the final part of this. So I've talked a lot about the fact that the chemistry is complex and not well understood. So we're going to talk about the models we use to look at understanding nutrition. There are three broad categories of experiments we can do: in vitro studies, in vivo studies, and more recently in silico studies. Each study type has pros and cons, so we'll mostly focus on the first two models here. You'll notice that these Latin words should always be written in italics, so just like et al, for example. Right, so this table summarises the difference between the two molecules, and this link here actually provides a good review of the issues. While there are significant issues with in vitro studies, that is studies done in the lab, in test tubes, hence in glass, in terms of their applicability to whole organisms, in vivo studies may also have problems. For example, the therapy can offer a short-term benefit but may cause long-term harm. But the two distinctions are here: in vitro in the lab, in vivo in life. And as you can imagine, doing in vivo studies is significantly harder to do. Animal studies and clinical trials are two examples of this. This looks a little bit complicated. This is just an example of the sort of way in which we measure antioxidants. So it looks quite complicated, but it's a straightforward lab analysis using dipycryl hydryl assays, which is this molecule here, DPPH. It's a free radical. It's naturally a free radical, indicated by the dot there, with its radical electron delocalized, that is to say, spread out over the whole molecule. So a big aromatic system. In ethanol solution is a deep violet colour. When it reacts with an antioxidant, it grabs hydrogen atoms from the antioxidant. This removes the radical electron, leaving a colourless product. So we can measure the amount of antioxidant effect. The reduction of intensity of the violet colour depends on the concentration of the antioxidant, making estimation possible. There are many other in vitro methods available, such as iron peroxide assays, Trolox, ORAC, estimation of nitric acid. See the paper cited in the notes if you're interested. You want to have a review of these. You may be thinking about a project sometime in the future. In vivo methods. For all in vivo methods, the samples to be used are tested or administered testing animals, mice, rats, etc. at a definite dosage regime as described by the respective method, so a certain amount over a certain period of time. After a specified period of time, the animals are usually sacrificed. Blood or tissues are used for the assay via the analytical methods outlined in earlier slides. And as mentioned, there are a wide range of potential analytes. Methods are reported for humans, but they seem to be mostly concerned with validating the methods rather than looking at health outcomes, which is a big distinction. Obviously, it's interesting, the science of how things work in terms of the detailed biochemistry, but what we're really interested in is, can we make people healthier? And it's very much an open question, and a question which we don't necessarily know how to answer. Okay, I mentioned in silico. Here in silico refers to biological experiments carried out entirely in a computer. This is a relatively new approach. It's been used in studies that predict how drugs interact with the body and potentially with pathogens. The main issue is clearly establishing how any computer simulation can model the epic complexity of living systems. Bacterial sequencing using PCR is well established, and the modelling of some molecules, for example, to understand drug mechanisms, is rapidly developing. In one example of whole cell simulation, researchers have built a computer model of a bacterial cell that, in a test of its response to sugar in the environment, accurately simulates the behaviour of living cells with respect to a small component of what cells do. Again, how well this relates to wider considerations is an open question. Which brings us to the difference issue with reductionism, problems with reductionism. The dominant approach in nutrition research traditionally is reductionism. The underlying assumption is that if we understand the details of parts of the issues involved in nutrition science, an understanding of the whole will develop. So this would be, for example, understand the detailed biochemistry of an antioxidant molecule. This approach has produced much valuable knowledge, but we're still a long way from understanding the relations between diet, life, and health style. Diet, lifestyle, and health. And there's a quote there from Campbell: "Such studies often create more confusion than clarification," and this is the sort of thing you often see in the media. this study says this, a different study says something different. Probably because people aren't reading the studies in the detail they should be reading them. So here's a couple of examples. These are among the headlines produced by 2 studies published in Lancet. They are somewhat contradictory and typical of much of the reporting on nutrition. In large part, this is due to the increasing need for researchers to get noticed and the tendency of their employers to promote such studies in the media. This is fine in principle, but often results in complex issues being oversimplified. That few news organisations have qualified science staff in their reporting teams these days only adds to the problem. So you can link through the original papers there if you're interested. Here's a summary of them. Despite the headlines and these two papers, the recommendations of these papers do not differ much from existing UK nutritional guidelines, as we can see from this table here. UK government recommendations on nutrition have been fairly constant over the years, reflecting the state of nutrition knowledge. There's a nice infographic linked here, which is worth having a look at. It's a reasonable summary of the issues, which I've summarised in this little table here. Read it and think about the points that it raises. Okay, so here's a possible approach. A meta-analysis is a statistical analysis that combines the result of multiple scientific studies. Meta-analyses can be performed when there are multiple scientific studies available addressing a particular issue, with each study reporting measurements that are expected to have some degree of error and which can be compared to each other. This pyramid is often presented as a hierarchy of improving quality in research. There are some issues with it, especially with respect to randomized controlled trials, which often may be very reductionist in the approach. They might be done on humans, but might be on a small number of humans, and perhaps people in hospital with a particular condition. How generalizable they are to wider studies is an issue. Remember we talked about generalizability earlier on in terms of blue zones. This is a sort of wider issue. Now, meta-analysis is a process where individual research papers on a specific topic are grouped or analysed and the overall conclusions drawn from the data they present. It's a specialised approach carried out by qualified teams of researchers familiar with their relevant areas. The Cochrane collaboration is a well-regarded international network with its headquarters in the UK. Its main goal is to produce high-quality, relevant, up-to-date systematic reviews and other synthesised research evidence to inform health decision making, and it does so in plain language, and that is one of the great things about it. So see some examples on the next page and have a look at the Cochrane collaboration video here, which I strongly recommend having a dig into Cochrane. There's lots of useful information in this. So if you want to know about vitamin E, you can search in Cochrane. It'll give you the latest information, what studies are saying, and present the result reports in plain language, which again is one of the key things which we're interested in this course. Again, there's an example, antioxidants for preventing and reducing muscle soreness after exercise. There's an example with a video, a short podcast, I think the chap talking about it. That's all, folks. That completes antioxidants. Thanks for listening.